California. Okay. Good morning, everyone. I hear there's a lot of people that are logging in from all over the country. So it's wonderful to have you all here today. My name is Sudha Rajamani, and as both Praveen and Neja mentioned, I'm associated with the biology department at ISRO Pune. I'm going to today introduce you to the science of astrobiology, which mainly deals with the origins, evolution, distribution, and future of life in the universe. And towards the second half of the talk, I'll also give you some flavor of how do we think about searching for life beyond Earth. So in my lab, this particular program is possible because of a whole bunch of people, PhD students, a postdoctoral fellow, a whole bunch of master's thesis students, and also several undergrads who do semester projects with us. And together as a team, we're very interested in understanding how non-life transition to life on the early Earth. And by the end of this talk, the hope is that you will also appreciate the context in which we do our work and the questions that we ask specifically in this particular context. So when do we think all of this happened? Here is a slide that gives you a sense of the early history of life on Earth. And roughly about 4.5 billion years ago, you had Earth formed, and a stable hydrosphere is thought to have come about by about 4.2 billion years ago. And between 3.8 and 4.2 billion years ago is when this grand orchestrated chemistry drama happened, which allowed for the formation of fundamental biomolecules relevant to what we see in biology today to have come about. And there are a few theories about how during this period of 4 to 4.2 billion years ago, the prebiotic soup was full of interesting molecules that is relevant, some of which is relevant to biochemistry. And over a couple of billions of years, you had this transition, or several hundred millions of years, you had this chemistry transition to biology on the early Earth. And this concept of transition from non-life to life or chemistry to biology is well understood in the next slide, where I'm showing you that there is this continuum that exists between physics to chemistry to biology. So let's take a molecule which is in the bottom panel. It's called cytochrome C. It's a protein. You zoom into it. It is fundamentally made up of single molecular entities called amino acids. These amino acids like glycine, alanine, serine, glutamic acid that you see here on the lower panel come together like pearls, and they form a string of pearls, which then suddenly now can do things which you cannot attribute to the independent monomers. Now zoom into one of these glycine molecules. What do you see? You actually see that it is made up of atoms like carbon, hydrogen, oxygen, and it has, glycine is one of the most fundamental of amino acids. But if you sort of get into more complex amino acids like cysteine, you even have atoms like sulfur. If you sort of get into a molecule like glutamic acid, it has a functional group on the side chain, which has a secondary carboxylic acid. So a lot of interesting possibilities here, which comes about because a bunch of atoms in the realm of the physical space come together to form these smaller molecules which exist in the chemical space, which come together and undergo a very interesting process called polymerization, which now allows them to do things which otherwise the monomers couldn't do with the same efficiency or just cannot do at all. And this particular phenomenon is called emergent properties. And this endowment of emergent property on a molecule is what we think allowed for the transition from chemistry to biology. And how do we think this fundamental biomolecules, the monomers, came about on the early Earth? It's thought that in two different ways this might have been possible. One is exogenous delivery. A lot of the previous talks that you would have heard as part of the START program would already allow you to appreciate the fact that there are a lot of these extra planetary objects like comets, meteorites, which are present. They can bombard the Earth. They do it even today, but it's thought that way back during the early history of life, there was a period when this bombardment was very, very rampant. And along with this bombardment and these pieces of extraterrestrial material, you actually have molecules riding on them, which can then be brought onto the Earth, like amino acids, like fundamental components that eventually form nucleotides, fundamental components that led to the class of molecules called lipids. And this process is called exogenous delivery. Another means of having very interesting monomeric molecules on Earth is possible because of really fun chemistry that can happen in certain specific niches of the early Earth. And some of these niches are thought to be like the ones that you see on the right panel here. I'm just going to turn on my laser pointer. This panel here is showing you an illustration of what is called a terrestrial volcanic geothermal pool, which exists at the edge of a volcano. It's very interesting because chemically and thermally, there are a lot of cool fluxes that happen there, which allow for thermodynamically uphill processes to be facilitated. Please understand that life is a far from equilibrium process. If you're at equilibrium, you're dead. So you need to be far from equilibrium. And for that, you need a lot of energy. Without energy, there's no life. So those kinds of energetic niches are allowed to have, are thought to have allowed for very interesting chemistry, which resulted in a whole bunch of fundamental monomeric molecules. I'm just showing a very small subset here, which is then used to transition from chemistry to biology or non-life to life. And the prebiotic soup is thought to have been full of very interesting molecules that interacted with each other and eventually formed the most fundamental primordial entities, which essentially connects all of these living entities that I'm showing you here on the screen. You have a mushroom here, you have the famous T-Rex here, you have the great Nelson Mandela here, you have a single-cell bacterium, you have a giant catfish, you have very huge trees called giant sequoias. They belong to different sort of kingdoms of life, but what is it that holds them all together or binds them all together? It's the least common denominator. It's called cells. So one way of understanding the problem of transition from non-life to life is to try and understand how the fundamental molecules in the prebiotic soup came together, interacted, and formed the oldest of all cells. So the way we think this might have happened, and there are a few different hypotheses that exist out there about how life might have originated on Earth. And this is one of the very systematically thought out hypotheses that allows us to in a very sort of stepwise manner, using tools of science, answer the various sort of smaller steps that leads to the grand problem. What do I mean by that? You first have to understand the prebiotic chemistry that led to the formation of building blocks. And I might have already convinced you that these building blocks are plausible in two different ways. Once they were present in the prebiotic soup, the next question to ask is, how did they go from this monomeric space to a polymeric space? Because that is where you now have cool properties emerging. So when I mean a non-enzymatic polymerization here that leads to the emergence of polymers, what I mean is you do not have proteinaceous enzymes because they're super complex. If you think about how a proteinaceous enzyme might have helped lead to all these processes, you're going to get into a rut. And that problem is called the chicken and egg paradox, because then you have to wonder about which molecule came first. So if you step away from two complex molecules and think about simple, plausible chemistry, you can now still invoke methods and processes by which you can think about monomers went to polymers. And once you had some of these interesting polymers, for example, like nucleic acids, like your RNA molecule, right, or a DNA molecule come about, some of them could have evolved functional capabilities to catalyze certain reactions. So we are very interested also in understanding how some of the functional molecules in the space to the formation lead to sort of their sustenance and their development to eventually interact with both primordial membranes. What are all of your cells made of? Your cells, my cells, every living being cells. They are predominantly made of a molecule called phospholipid. We don't necessarily think it was a very complex bilayer very early on in the primordial cell or the protocell. It might have been a simpler version of these phospholipids called single acyl chain fatty acids. But this crosstalk between the informational polymer like a nucleic acid and the membrane would have set the stage for the emergence of the protocells, which also would have had to encapsulate some sort of minimal proto-metabolic network to allow it to use energy from the outside, live, grow, and sustain. So this is sort of broadly the hypothesis that we kind of keep in mind when we are thinking about how we think about the various steps of this transition process. But look at life today. What does it have? It has this enormously complex cells, right? Some of the oldest cells that are thought to have emerged close to 3.8 billion years ago. One of the oldest evidence is about 3.8 billion years ago now, is called a prokaryotic cell. And the example of it is a bacteria. Even though this cell looks simpler than the eukaryotic cells, which make up you, me, the plants and higher order organisms, it still is super complex. And to think about how this magically emerged from the prebiotic soup is completely non-trivial. Hence, we think about protocells as entities which literally have a bilayered membrane. So think of a tennis ball. The outer yellow layer is a bilayer lipid membrane, and take a slice of it off and you're looking at the interior of it. You actually have an encapsulated genetic molecule of some sort, something which encodes information, given that all of life now encodes information in the form of nucleic acids. The most prominent class of information coding molecules that we think would have existed even back then are nucleic acids, but RNA is the winner here because RNA can do two fascinating things. RNA can not only encode information like it does in the SARS-CoV-2 virus, but it can also catalyze reactions. It can fold back on itself and form these enzyme-like pockets which allow for very interesting catalysis to happen. So we think that these protocells, or this lipid membrane-bound minimal living systems, at some point led to a more complex ancestral community of cells, which then eventually led to your prokaryotes. So the tree of life, a sort of a minimal cartoon illustration version of it, which you know of or which might exist in some of your textbooks, alludes to this last universal common ancestor as being something that came about, which then led to the three domains of life. Now, this is a bit of an old story. In the last 20 years, the work that we are carrying out and many other colleagues in the field are carrying out is giving a different picture. It seems like there probably was a common ancestral community of primitive cells or primordial cells, which interacted with each other, just like the cells in your tissues of your body interact with each other. And the selection pressures or the physicochemical parameters of the planet that existed then allowed for a subset of them to become victorious, which then led to the three domains of life over several hundreds of millions of years after that. Why are unicellular organisms so fascinating? In addition to being the first living entities that might have formed on the early Earth, they are just mind-blowing in a way that you and I can never be. They basically can populate every extreme niche that you can think of. There are highly UV-resistant organisms. There are hyperthermophiles that kind of love extreme heat. There are halophiles that can survive in extreme high concentrations where you and I will shrink and die. And there are permafrost-dwelling organisms where you can have organisms which can tolerate very, very low temperatures. These are called cryophiles. So unicellular organisms might not be complex in the way we think of multicellular organisms like you, me, plants, fungi, but they are nevertheless ultra cool, and the small size and several important properties allows them to do things which we can only dream of. So given the super importance of unicellular organisms, we try and understand how their ancestral versions or protocells came about. And we think about, and when we think about where these amazing things might have happened, you need to think about environments that would have allowed for this interesting chemistry to biology transition. Depending on whom you talk to in the field, there are two famous locations. One is the hydrothermal vent system, which is a volcanic system that exists on the floor of the ocean. The other system is a terrestrial geothermal or tidal pool-like system, which exists right next to volcanoes on terrestrial environments. Here is a picture of a hydrothermal vent called Black Smoker. It kind of exudes black smoke because of the chemistry that is possible in the rocks there, because of the nature of the rocks and because of the way it interacts with the water. Whereas in a terrestrial geothermal pool, you see kind of these white smoke coming out. A predominant amount of the smoke is fundamentally carbon dioxide. In addition to that, you also have hydrogen sulfide spewing out, which then interacts with water to form sulfuric acid. So whenever you go to a geothermal pool, not all geothermal pools are equally good to go and kind of put your finger or your hand in or yourself in. You could actually completely burn yourself in some of these pools because of extremely acidic pHs. There's a whole range of pH and temperatures that are possible in these geothermal pools. So be careful when you sort of go and see a geothermal pool and get super excited. Don't please put your finger in. No? First check, take a pH strip with you and check what the pH is first. Yeah. Sorry.
You can hide it. There is a message on your screen. Down.
I don't have any message. Oh, this thing here. Oh, down here.
Yeah,
okay.
Yeah.
Hang on, 1 second. Let me. Sorry about that. Yeah. Is that good? Hello. Is that good? But I
have it and... fine.
Okay, fine. Okay. So let me go back to pointer. Yeah. So in these environments, we basically think a lot of interesting chemistry and minimal biochemistry was possible. But when did people even start thinking about this in such a systematic manner? It goes back to early 1920s. This is the time when the famous operant and Haldane theory was put forth, which essentially posits that if you had fundamental inorganic molecules in your primordial ocean, like ammonia, carbon dioxide, water, methane, hydrogen, dihydrogen, and if you had a nice energy source like ultraviolet radiation of the sun or lightning in early Earth atmosphere, you have the right kind of environmental conditions that allow for the transition of this inorganic molecules to organic molecules in a spontaneous manner. You can actually go from this very reducing environment of molecules to a space where you can now have fundamental ingredients of life like amino acids, hydroxy acids, urea. Yeah. So they basically proposed this theory. The two of them didn't know each other. No internet back then. Letters would take 3-4 months to get to each other. So independently, the two of them were sitting in 2 different countries and proposed this. A theory is a theory till it is actually demonstrated using an experiment. The experimental evidence for the operant Haldane theory came about in 1953 for the first time. If you all remember, 1953 is the same year DNA's double helical structure was described. 1953 is also special for other reasons, which you need to go back and look up. But it's also the year when Miller and Urey, Stanley Miller and Harold Urey got together, and Miller did the very famous spark discharge experiment. Sorry, some of the words here look weird, but this is essentially methane, this is ammonia, this is water, this is hydrogen. He added these ingredients to what was supposed to be simulating a primordial ocean. And in this setup, which is actually shown here, this is the original setup that Stanley Miller worked with. He basically used high temperatures. Early Earth is thought to have been a very harsh space, not as beautiful and conducive as it is now, temperature-wise. Unfortunately, temperatures are getting hotter as we speak, but it was super, super hot, right? So he was simulating those very early Earth temperatures, and this water contained all these different reducing these molecules which are simulating a very reducing environment. And on vaporizing, they go to this space where you can actually have a simulation of lightning, for example. He basically used an electrode to simulate the lightning, and this is of much higher energy. And whatever happens, fun reaction happens here. Then the fumes go down this condenser. The condenser has cold water flowing around it, so gas condenses into liquid. And once it comes down here, you can use this little stopper here to collect your samples. So they did this experiment. Miller did this experiment for multiple days. I am told even for months on end. And what he showed with very, very simple techniques which were available back then, that you can go from a bunch of these inorganic molecules to molecules that make up your life molecules today. So this was the first proof that you can go from non-life to life. And I showed you 2 different niches where it's thought that such interesting chemistry is possible. In our own cool lab here, in the Chemical Origins of Life lab here, we simulate terrestrial hydrothermal systems, and we try and understand how can you go from a bunch of independent molecules to protocellular systems. And there is a model in which which we factor in, which is based on logical deduction, which is based on knowing the geological settings of the early Earth, some factors of it, which is based on knowing how compartmental systems like vesicles, like small cells, behave under these conditions. So essentially, if you start with monomers of, for example, your RNA molecule, which is called ribonucleotides, and if you start with the monomers of a lipid, a bilayer forming, a membrane forming molecule. These, a quick word about these membrane forming molecules. They are amphiphilic in nature, which means they have a water loving and a water hating group. So the second they see water, the water loving groups will tilt towards the water, and the water hating groups will go away from the water interface, which is what you're seeing here. It's a lot like your soap. The second your soap hits water, what does it do? It starts bubbling into these nice bulbous balloon-like structures, right, of various sizes. Very similar kind of behavior. So if you have molecules of these two fundamental biomolecules in the same tidal pool, and you can now think of geologically plausible wet-dry cycles. Wet-dry cycles, like they happen on today's Earth, they would have happened much more readily on the early Earth. It is driven by either seasonal changes, day-night cycles, and I already told you that early Earth was much hotter than now. So at daytime, hot conditions, all the water in this pool evaporates. Nighttime, things get cooler, you can have condensation, and the water in the pool comes back up again. So in this kind of a cycling environment, if you had fundamental monomers of both your nucleic acid and the bilayer present in the same space, you can have interesting reactions happen now. What do I mean by that? Already the lipid molecules form a lot of interesting higher order aggregates, but let's focus on this thing called vesicle. The second, the vesicle is in a space where the water is removed. It collapses into these lamellar structures, and every alternate layer has water loving head groups, where if you have other solutes that are also polar, they can now get constrained in two-dimensional spaces where interactions are much more readily possible. The insert that you see here is actually a image, electron microfreeze fracture microscopy image of some of these lamellar lipid bilayer structures. So you can go now to from a three-dimensional space to a two-dimensional space where you're constraining molecules closer to each other, you're increasing their effective concentration in a smaller space, and you're facilitating interesting collisions between them, and you're also pulling out water. So what is readily possible in these conditions is what is called polymerization. Polymerization is essentially a product of condensation reaction, where you're pulling out water molecule. You can essentially stitch to amino acids using a peptide bond under such conditions. You can stitch to nucleotides into a dinucleotide or polynucleotide, which is connected by phosphodiester linkages by pulling out water. So depending upon the starting solute, you have lots of interesting possibilities, but let's assume these are ribonucleotides. Every dehydration cycle, you're facilitating polymerization, and every rehydration cycle, I told you, the second the lipidic molecules, the amphiphilic molecules hit water, they are going to bleb out into bulbous structures. You see this microscopic image here? This is a real image of a very simple fatty acid system made up of decanoic acid and decanol, a C10 hydrocarbon chain containing system. It very readily blebs out into these tubes and into these balloon-like structures, and it takes any kind of sort of polar molecule along with it, and this very green fluorescent color is actually a polar dye, which is giving you a sense that it is getting encapsulated within. So we think a similar process is what would have led to the first protocells, and multiple rounds of dehydration, rehydration, and some form of selection. Maybe some processes gave advantage to this vesicle versus this vesicle, which is why this vesicle is breaking up and dying off, and this dying off in quotes, and this vesicle is surviving and continuing. So it's thought that this kind of process is what is the main way in which you went from the prebiotic soup of molecules to protocellular systems. So how do we now think about the informational polymer evolution? A quick word about that. Again, using RNA here as an example, what you see here are the two nucleic acids that we see in our cells and in living systems now. RNA, this is kind of the molecule which encodes information in many viruses, RNA viruses, including SARS-CoV-2. Though DNA is the main molecule that encodes information in your cells and my cells. Very early on, though, it is thought of to have been a much, much more simpler molecule, because forming these molecules directly from the prebiotic soup is completely non-trivial. And that is something we have learned the very hard way by systematically doing these experiments. So now we are thinking about even more simpler molecules like pre-RNAs and proto-RNAs, where the fundamental logic stays, which is you have a you have essentially a moiety, which is like the nitrogenous base here that allows for recognition and interaction with another strand of nucleic acid. You have a trifunctional connector here, like the sugar, ribose or deoxyribose, which allows you to connect 3 different entities together in a unit. And you have an ionizable linker like the phosphate here. It could have been any other kind of linker. We do not know. So what is very important for you to remember is this whole prebiotic soup that I kept talking to you about. We still remotely do not understand the complexity of the soup. There would have been many kinds of molecules that would have been plausible. While life shows what it did, or how did molecular evolution result in the use of molecules that we see in life today, is still something that we are trying to systematically understand. But in my own group, we actually are trying to look at some of these pre-RNA molecules and try and understand what other kind of interesting synthetic nucleic acids can you actually work with, both in the realm of origins of life and for use in biotherapeutics and so on and so forth. And the other important thing to think about is the membrane itself. Again, it might have not just been made of one kind of molecule, right? You can very readily have a whole set of amphiphiles present in the prebiotic soup. So one way to think about what were the winners and what did not survive is to invoke some of the physicochemical parameters that would have been hugely important in shaping the space, just as it does today. Only thing is, a lot of biochemistry that happens in your body today is happening at very ambient temperature and pH conditions. But on the early Earth, all bets are off. It could have happened at a whole bunch of different temperatures, or at a whole bunch of different pHs, depending upon the physicochemical parameters that is constrained within a certain pool. And pools are very interconnected. If you go actually to, if you go and visit like Yellowstone National Park, you probably will see hot springs where they're present next to each other. They seem separate, but they're connected below the earth with sort of, they're connected below the earth in some sort of way. So there's, or even actually you see runoffs even on the surface. So there's some sort of connection. So one pool, which is at pH 4, can have one kind of interesting chemistry happen. Another pool with another sets of conditions can have other kinds of interesting chemistry happen, and they could mix in some third pool and result in more interesting possibilities, or it could die off. We do not know. So it's extremely important to think about the kinds of molecules that eventually led to the formation of these robust membranes, because I told you earlier conditions were harsh. So very early on, if you worked with simple molecules have stability issues and other kinds of problems, but at the same time, you mix a couple of simple molecules together, they can start doing very interesting things. So we are very interested in also understanding in the field what are these membranes, or membrane molecules, and what might have been really good protocellular systems. And once the earliest of cells came about, it's important to also start thinking about how they might have evolved to more functional cells. When I say functional cells, these are much closer to cells that we see in your body and my body, where you have protein channels in the membranes, which allows shuttling of certain molecules across the membranes. You have other kinds of channels which allows for transport of ions, which is extremely important for metabolic processes. So these are also important factors to keep in mind when you're thinking about this emergence of protocellular system, right? And if you're now thinking about a metabolic network that allowed for the system to exist far from equilibrium, one way of thinking about this is to think about minimal enzymes, which are not as complex as your modern enzymes, but you strip down the complexity. At the core, you see such scaffolds, for example, porphyrin, like you see in hemoglobin. One could simulate some of these scaffolds, or even go back just to the hydrated metal ions and try and understand how much their functional capability changes when you kind of strip off all the complexity, and see if you can work your way up to form some of these minimal enzyme complexes, which can also be incorporated to facilitate some minimal metabolism in this protocellular system, all of which basically allows it to survive, grow, and reproduce. So finally, it's also crucially important to think about the multiple crosstalks that can exist between all of these processes, because they all have implications for the protocell formation. So how do we do this in the lab? We either set up experiments. No, this is not me, or maybe this is me, I don't know. We basically set up experiments and hope that at some point, we'll see the oldest, of course, we're not going to see even cyanobacteria come about, but primitive cellular or primordial cellular systems which behave lifelike. The other thing to also importantly do is to go out into planetary analog environments. Here you're seeing 3 such hot springs. They don't look as cool as you would imagine them to be, but trust me, they are very geologically interesting, geochemically super interesting. These are in our own Ladakh, and you can go out there, come back with the hot spring samples to the lab and use them to do your experiments in a much more realistic manner, or you can get permissions and then take your own experiments out there and do some small kind of experiment out there to better understand how your reactions of interest actually pan out under natural conditions. So suffice to say, in these 6 or 7 decades since 1950s, we have understood a lot of prebiotic chemistry, which has allowed us to appreciate how you can go from this inorganic molecular space to molecules that form proteins, molecules that form nucleic acids, and molecules that result in membranes. So are we close to using a bottom-up approach to synthesizing life? The answer is, I don't know, maybe, but very importantly, we are much, much, much better off to draw up a story which is consistent with real evidence from geology, from chemistry, and also from our understanding of extreme life, the unicellular life which populates a lot of niches on the earth, to basically try and fit the pieces of puzzle together to eventually understand how maybe life originated on Earth. And why is all of this understanding super important? It is super important because if you want to ever look for the presence of life in the universe and understand if there's life out there, you need to at least understand what is tangible. And the most tangible processes is happening on your own planet. You can go to different regions of your planet, study the geology, understand the geochemistry, do very interesting experiments from the logical deductions that you sort of derive from this various understanding. And once you have figured this puzzle out, and as you figure this puzzle out, it systematically informs how you can think of whether there is life out there in the universe. And how many of us have not thought about this? Thanks to Steven Spielberg films, we imagine this life to be a very complex organismal creature, which is probably taller than us, has much bigger heads, like the aliens, right? Maybe the aliens out there are not really out there and then kind of looking at you back, right? They might just be microbial in nature, because like I told you, single cellular entities can survive. If one day just like the dinosaurs, you remember when the dinosaurs were present about 200,000 years ago, there were a few cataclysmic events that happened. They wiped the dinosaurs out. How do we know that they exist? Because of signatures that they left. They're called biosignatures, in this case, fossil signatures, right? We knew that they existed. They got wiped out, but the rest of the smaller organisms and the microbial organisms survived, which then allowed for the evolution of a mole-like ancestors of us mammals. And what we see today happened in the last 200,000 years, which is, if you take a 24-hour clock, the original drama began 4 billion years ago at just after 12. The rest of the drama went on for several billions of years. And this multicellular species, very complex organisms, appeared only around 2 billion years ago. And we appeared much, 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 much later, very close to midnight. So it's probably very important to think of, when you're thinking about looking for presence of life in the universe, to think of microbial entities, because it's much more readily detectable if it's present in large numbers. And is there life out there? We don't know yet, but the good news is that it might be possible with this crazy amount of exoplanet discovery that's going on right now, which is, exoplanets are essentially planets, but they're present around other stars. There are a whole different set of exoplanets, right? Some are very much Earth-like. Some are much, much, much larger than Earth, and they're called super Earths, and they're rocky in nature, which is why they are Earth-like, yeah? Then you have Neptune-like exoplanets. Then you have gas giant-like exoplanets, which is similar to more similar to Saturn and Jupiter. You have this whole plethora of planets out there in the universe. Some of them harbor the kind of conditions which might just have allowed for the emergence of life, or what we think of as life. And this is possible if it exists in this zone called the Goldilocks zone, which is the space around the star, in the orbiting space around the star, where the conditions are just right. It's neither too hot. Mercury, super hot. Venus, very, very hot. The atmosphere, the surface atmosphere of Venus is 400 degrees centigrade. Earth, ambient. Go out to Mars, already cold. Go further out, extremely cold, icy worlds. Outer planets are all very icy and extremely cold, right? So this habitable zone, which is the perfect distance from the star that allows for the planet to be able to harbor liquid water and have the right atmosphere, because only a proper atmosphere will allow for liquid water also to hold. This set of planets are what are extremely interesting when we are looking for life. And they are argued to exist in what is called this Goldilocks zone, where it's neither too hot or not too cold, and it's just right. And finding these just right planets, it has been the mission for a whole bunch of space missions, particularly the very cool Kepler mission that's happened, and the new test mission that's going on. But please also remember that just because a planet seems to be habitable, that doesn't equate to signs of life. A habitable planet is very interesting for us to peer at and look at from Earth, or send missions out to understand whether they actually harbor real signs of life. And how do we understand these signs of life? Signs of life are mainly sort of, for example, if you're going on a hike, right, and you see a leaf, maybe there's not a tree in the immediate vicinity, but you know that it came from some plant or a tree. You see a feather, don't see a bird in the very immediate vicinity, but you still know that the feather came from a bird. So these are all examples of biosignatures, which either could be elements, molecules, substances, patterns, structures, characteristics of all of this that indicate either the presence of current life or presence of past life. And when we are talking microbial life, we are predominantly looking at elements and molecules as biosignatures. And very, very interestingly, these biosignatures have to sort of be large scale, because if they're not present in reasonable abundance, it's very hard to detect them, either from using very cool high-end telescopes from Earth, or by sending out flyby missions to planets to look at them, for example, or to moons. So one other very important thing, look at Earth today. This pale blue dot, as the very famous Carl Sagan called it, looks the way it looks today because life has transformed the planet. Very early on, when there was life just beginning to come about, or there was just no life. This is an artistic rendition. We do not know if this is how it looked, but logically we can assume this is how it looked. It's supposed to have been, Earth is supposed to have been an extremely hot planet, yeah? When life came about and sort of just became so much more rampant, it completely changed the planet's atmosphere, the planet's surface, the planet's water, the environment, to the point where your planet's appearance completely changes over time. So that is also something which is very, very crucial when you're thinking about biosignatures and looking at a certain extraterrestrial body. And biosignatures of a particular body, a moon, an exoplanet, always must be assessed in the context of that planetary environment, because the geological history of, for example, Venus is very different from Earth, is very different from Mars. So it's also important to understand the geology and the mineralogy of the planet, and then assess the biosignature accordingly. Otherwise, you might be making wrong interpretations. What are the biosignatures and life detection methods that are out there? A whole bunch of them, because depending upon your signature, whether it's at the atomic level, elemental level, molecular level, polymeric level, or at the cellular level, or at the level of much larger patterns, you can use a whole bunch of detection techniques depending upon the size that you're working with. For example, you can use X-ray based methodologies, mass spectrometry methodologies that allow you to understand atoms. And you can basically look at infrared spectroscopy, use Raman spectroscopy, NMR to look at molecules. You can use more sort of robust mass spectrometric and chromatographic methodology to understand more complex polymers and higher order structures that make up your cell. You can use electron microscopic methods to look at, you can, when you look at under the microscope, electron microscopy is something that whose resolution is very, very large, and you can see at the molecular interaction level things in detail. And then you can actually have at the much more complex levels, like if you have a satellite, you have satellites actually going around the Earth, which are geomapping Earth. And just like that, you can essentially geomap another planet which is close by to wherever you can send out one of these satellites. And basically, depending upon the scale of the biosignature, you can use a whole bunch of detection techniques which allow you to be able to detect them. And some can just be visual observations, right? If you have a meteor fall, and this happened, I think, as recently, I don't know if it's still fully delineated as a meteorite, but it fell in Trichy in Tamil Nadu, and you can actually visualize the meteor, right? Then you can take the meteor back into the lab and you can drill to the core of the meteorite, extract the organics and the aqueous phase molecules, and you can use a whole bunch of these techniques to zoom into it and look at whether there are interesting molecules there which are relevant to life. So biosignature detection, very possible. Biosignature assignment, very non-trivial. This is something that scientists across the world, including in our own country, are trying to systematically do in order to be able to confidently call out a signature as a biosignature. So what are the few ISRO missions that have happened in the past, which basically have the potential for doing astrobiology related observations? The very famous Chandrayaan series of missions. Chandrayaan-1, for example, actually for the first time showed the presence of water on Moon. Then you have the Mars Orbiter mission, which another advanced version of it called MOM-2 is going to go. There's going to be a lander, there's going to be a rover, and there's a whole scope for very interesting astrobiology related experiments if ISRO has astrobiology as one of its foundational ways of thinking about some of these missions. There's a planned mission to Venus. Venus came in, suddenly became famous in the news in the last couple of years when phosphine was observed on Venus. I don't know how many of you remember this, and there was this huge debate of whether it came from a biological source or from a geological source. Without doing systematic research, one can never be sure, because phosphine is very readily produced by geological activity, but on Earth, a lot of phosphine actually comes from biological activity. So how can we discern this for sure? By, for example, on this planned mission to Venus, say, send some of these spectrophotometers as mentioned here, which during a flyby can actually sort of suck in some of the gases in the outer atmosphere and actually look at it systematically using these extremely high-end spectrometers to understand whether it has any molecules relevant to life. You can essentially also think of a lander. A lander is a much, much more difficult problem because your clouds of Venus, where we think things are more ambient and life might be possible, we do not know. That itself is extremely acidic in nature. So you have to, if your lander has to go past this environment and land on the surface of Venus and survive both the impact and send a rover out to move on it, it has to deal with extremely high temperatures, a lot of acid corrosion. So is that technology possible? I will say it's plausible, and a Venus lander experiment is in the works from other countries. However, it's going to be completely non-trivial. And then we also have this planned LUPEX mission, which is in collaboration with JAXA, or the Japanese Space Agency, to explore the lunar South Pole. So a lot of interesting possibilities for astrobiology related experiments. And we are very excited about collaborating with ISRO on this. And we'll leave you with what is happening now as we speak. Mars 2020 Perseverance rover out there on Mars. It landed in Jezero crater. The reason they picked Jezero crater is because of its mineralogy, because of the conditions that exist there, and because of signatures of water that happen to be present on Mars. They felt that if you're looking for any biomolecule or ancestor of biomolecule present, it is possibly one of the places on Mars where you could much more readily detect it. So the particular, rover has a whole bunch of very high-end spectrophotometers, all of them predominantly trying to understand the mineralogy of the planet, which tells you about what elemental composition it has. It's trying to look for organic compounds. It's trying to even sort of, there are cameras. This Watson camera is trying to actually map the geology, the surface of Mars, and understand the surface features much better. We have some idea about this because of several flyby missions and previous rovers and landers sending back very cool, interesting data to us, but we still do not understand all of it. So all of this hopefully will tell us if Mars ever harbored life in the distant past, because now it is geologically dead. But very early on, there is thought to have been tectonic activity out there, and presence of water makes it extremely exciting. So let's see, maybe we will detect biomolecules, if not even a microorganism, not sure. Extremophile, yeah? So with that, I will tell you that it's super fun to think about these questions. We don't work on all of it, but we work on a good subset of it. And this is the lab over the years. And astrobiology research is not just happening at IISER Pune. Direct astrobiology research is happening in a few different parts of the country. And one of the only centers for excellence in astrobiology is in Amity University, Mumbai, which starting this August, in a few weeks, is going to start an MSc course in astrobiology and space sciences. I'm very excited for the course and the possibilities. In an Institute of Astrophysics, a whole bunch of colleagues there looking at the cosmology and the astronomical aspects and astrophysical aspects of various processes, which is crucial for informing us about how do we think about early Earth conditions and solar system conditions. PRL Ahmedabad, and you also have colleagues from, in PRL Ahmedabad, we have a bunch of astrochemist colleagues. I think one of them even talked to you. So astrobiology, please remember that it's an extremely interdisciplinary area of science. And I forget where is that very, very important image that I always show. If you can give me 1 second, maybe I can pull it out of here. So this area of science feeds off of information from multiple other areas of sciences. Neerja, can you see this particular slide where I'm showing astrobiology and other areas of sciences? Awesome. Yes. So this is not fully all-encompassing sort of picture, people, but the idea is to say that the science of understanding the origin and the emergence, the evolution, and the distribution of life in the universe, not just on our planet, involves information that you can gather from several areas of sciences: chemistry, biology, geology, aspects that I've touched about. I didn't talk about astronomy, physics, paleontology maybe when I said fossils, right? But suffice to say, and math underlies a whole bunch of these sciences. So suffice to say, astrobiology is this extremely interdisciplinary area of science, which can use expertise from several different and diverse sets of people. And in our own lab, we use multiple tools in biochemistry, molecular biology, biophysics, organic and inorganic chemistry, geochemistry, and we collaborate with people who do experiments on computers or experiments on notebooks to model many of these reactions, because it is complex enough that all of it cannot be done just experimentally. So with that, I will stop and I'll say thank you.